Thank you,
Thank you,
guys.
Johan. Thank you, the chairs and Ellen and the MMF team to give us this time that has now become kind of a annual tradition. It doesn't want to be a lecture. It wants to be just a very brief overview, not comprehensive, about what we have available at the moment and where we are going as a future directions, and just to give you some insights and some food for thought. And we're really hoping to just stimulate the discussion for later. So you have heard a lot of Edinburgh, so I thought to put a picture of Glasgow for now. But I think that the first question we have to ask is, why do we really need cholangiocarcinoma preclinical models? And I believe it is important we keep this question in our mind if when it comes the time to decide how we want to use the preclinical tools available. Because there are different points that we need to keep in mind. First of all, not all persons from the general population will develop cholangiocarcinoma. We have heard it's becoming an increasing tumor, but still only 1 to 5, at most 10 persons every 100,000 people will develop cholangiocarcinoma in the Western world. So we need some models that can mimic this development. We know cholangiocarcinoma is heterogeneous in terms of locations, but also in terms of genomics. And we know that some tumors will remain confined within the primary site for a long time and can give the possibility to undergo surgeries, while others, unfortunately, can develop quite rapidly. And we also know that after surgery, some patients are cured. Some patients, unfortunately, will see their tumor recurring even after adjuvant treatment. As well as with the systemic anti-cancer treatment, we know the response can be different, and we need to be able to model and mimic these different types of responses. So we need the models to understand how cholangiocarcinoma develops, but we also need some tools to understand the biology, the differences in mechanisms of growth and spread, and obviously also to try and predict the drug response with a long-term aim of increasing the cure and the long-term benefit for patients. So just to give you a very brief overview of what are the different models that are available, we can consider some which are very simplistic. If you look at the top of the slide, you see some flies. They don't even have a liver, so they haven't been used extremely very much in cholangiocarcinoma, but they still represent a useful model if you want to insert genomic alterations. We have seen that some genomic genetic mutations are quite important in cholangiocarcinoma. And then they are very easy to be manageable, so you can increase them, you can test with drugs. So you can see the effect of these genetic transformations and study the effect on drug sensitivity and kinase and target engagement. We can increase the complexity of the model, going, for example, to fishes. They start to have a liver, but they are still very short-lived animals, so the simplicity of doing some experiments is there. Obviously, mice and rats, you have seen from the previous presentations, are the ones that are most widely used, and we will talk a little bit more about that. But we also have the possibility of going into larger animals. Non-human primates have been used for the study in liver, for example, and also more recently the oncopig has shown that not only is useful to reproduce the liver cancer, but also the background, for example, in modeling NASH, which might become also relevant in cholangiocarcinoma. If we consider some patients-focused model, which consider human-derived cells, we can again start from very simple model like the 2D cancer cell lines, and you have seen that in the previous presentations, so those are very easily manipulated, so we can use them to answer a lot of different questions. You've also seen that sometimes we can aggregate these 2D epithelial cells in creating spheroids. In this case, we are mimicking the gradient for example, drug penetration into the tumors, and we can then go up to cancer organoids, which are considered 3D self-assembled structures that derive directly from the patients. Obviously, the limit of the cancer organoids is that we are still considering only epithelial cancer cells, but we have seen that those are not enough to understand the biology of cholangiocarcinoma, and the idea and the future is that we can develop the assembloid, so we can include these epithelial cancer organoids into a structure that also has the microenvironment to have a better idea. And then obviously we have the tumor tissue slides we have here this morning. We have heard that those are less functional, but still quite useful for drug sensitivity. So if we start from the animal models, I'm not expecting you to go through all these slides, it's just to tell you that we have a different spectrum of types that we can use. And you have seen many of these in the presentations these days. Today, we have sometimes the possibility to give a carcinogenetics, a diet that can induce the transformation of the cancer. And some other times we want to focus on a specific genomic mechanisms, and so create a genetically engineered mouse model. We also have the possibility to inject human cancer cells into the mice, either under, the subcutaneously, as we have seen before, or within into the liver to try and replicate what happens in humans. But we can also inoculate in the mice the tissue directly coming from the patients. You've seen these slides before from Luke's team. We can use, for example, these genetically engineered mouse models to dissect down the effect of specific molecular alterations that we believe important in cholangiocarcinoma, and then we can use them to really assess the sensitivity to different compounds. This, I don't know how to make it work. Sorry, Luke, because there should be a video. If you can click on the picture, the gray picture. This is, again, another model that Luke's team has been using quite frequently in using hydrodynamic models where you can be more flexible in altering the gene expression within the liver. And I thought that this was quite important. Is a cost-initiated project led by Laura Faustier in France, and then with the co-leadership of Diego Calvisi and Luc Bolter, where they tried to put together all those different criteria which are necessary when we develop novel preclinical models. You have seen this morning that we can have a lot of different models that can probably mimic different situations, and sometimes we want to understand which is the clinical relevance of these models. So it's always important to understand which kind of situations we are modeling with that specific model. So I think probably this is one of the things we can discuss later. And moving on to patient-derived organoids, I'm pretty sure many of you have already heard about this new functional model. As you can see on the left of the slide, you have the possibility of growing the tumor in vitro, is what you might have heard in the press, like mini tumor. So taking a little piece of tissue from the biopsy of the surgical samples, you can grow them in the lab, and you can see that they are able to recreate exactly the same anatomy and morphology that the tumor shows and displays in the normal H&E. Obviously, when we compare with other patients focused models, they have some advantages and disadvantages. If we compare with the 2D cell lines, definitely they seem to have a more robustness in predicting drug sensitivity, and we'll see this one, as well as recreating and remodeling this cell-to-cell interaction and cell-to-matrix interaction. If we compare with the PDX, when you take the patient's tumor and you try to grow that in mice, these become less expensive, and you understand that there is not the need of creating the mice facilities, but obviously is lacking the tumor microenvironment that can be available in the mice. The organoids, when you grow them, you can test them, and now this is quite accepted. They can recapitulate the same genomics that is seen in the patients, but on the right of the slide, you can actually appreciate that even if you keep them in culture, they maintain the same genomic profiling. So this is becoming quite interesting to see how we can expand these organoids and then use them as a model that recapitulates that specific tumor. So the organoids can have different downstream applications. First of all is they give you the possibility to have a long-term in vitro expansion of the tumor. And this is particularly relevant in cholangiocarcinoma, where sometimes we don't get a lot of tissue from the biopsies of all the patients. So we have seen that a lot of biomarkers now rely on much more complex analysis that cannot be done only in some small core of tissue. So this could potentially have an application. You can also see that we can genomically manipulate the organoids, so this gives you the possibility of having a functional tool that you can modulate and you can use to dissect down the biology of the tumor. We can use them for drug screening, and we'll see this a little bit more in detail. But also the organoids have recently given the possibility to prompt a development of innovation and technologies. So not only in optimizing protocols for the development of organoids, but also trying to create a more automated system for high throughput screening, as well as co-culturing and interacting with specific 3D bioprinting of the liver, for example, to create what we said before we want to generate as in organs on chip. So recreate really a potential cholangiocarcinoma in plastics. So if we focus a little bit on drug screening, we have seen this has been our experience where we derived the organoids from an intrahepatic cholangiocarcinoma, patients who unfortunately had progressed to all the standard of line chemotherapies. And then we grew these organoids in plastic, and we have been able to put a lot of drugs on these organoids. So we could see that the same drugs the patient had become resistant were actually not, were not very active on the organoids, but you can still see some yellow spots identifying some drugs that can be potentially useful. In these specific projects, we were really interested in some, specific compounds and biomarker development, drug discovery, but, this could actually be taken forward more into clinical practice. And many of you might, already be familiar with the data by Nicola Valeri, the ACR Marsden, where in other tumor types, for example, this specifically focused on colorectal cancer and gastric cancer, co-clinical trials have shown that the organoids can actually mimic the response that the patients have in vitro. If you look at the bottom of the slides, you have those colorful lines identifying how organoids respond differently to a chemotherapy drug, and that is exactly the same chemotherapy drug that had been given to the patients. And you see that only the yellow line shows, a response of that, the organoids deriving from that patients who was only the only patients who was sensitive in vivo. So this gave us the possibility to say, maybe we can use the organoid like a real life screening. And if we think about introducing them within the clinical management of a cholangiocarcinoma patients, the idea is that we can add something on what we are doing now. If at the moment we use a genomic characterization on tissue or blood for informing a second-line treatment, the idea is that we can also derive organoids from that specific patients, and then we can give better information for the second-line treatment. So with the idea that we can move from a precision approach to really an individualization of the treatment for each given patient. Unfortunately, this is not so easy to be applied. If in colorectal cancer things are moving forward, we are a little bit behind in terms of biliary tract cancers for two main reasons. The first one is because we still don't know exactly which is the efficiency rate of cholangiocarcinoma organoid development, especially if we dissect down all the different subtypes, but also because to derive the organoids, we know we need to process the tissue fresh within 6 hours, and this requested an high-level technical skills, which means that potentially, not all patients going into different peripheral hospital can have these laboratories available. We also know that in biliary tract cancer, we need to merge the efforts together, and we can change clinical practice only if we do multicentric clinical trials. So I think that one of the things we wanted to see is how we can really increase the applicability of organoids into multicentric trials. So focusing on the efficiency rate of organoids from biliary tract cancer first, we don't have a lot of data. We do have experience and feasibility data. We know that single cases or small cohorts have shown a success, but what is the application on a routine standard is still unclear. A recent collaborative efforts between UK, Netherlands, and America has shown that apparently, as you can see on the first graph on the left, you have about 75% of lines that can be developed, but then the efficiency becomes less if you consider those who are truly malignant, even though I think we can discuss here how you really assess them. But it's also important to notice that it looks like the differentiation grade of the tumor, the degree of differentiation and the aggressiveness of the tumor doesn't really correlate with the ability of doing organoids. And you can see that there was an effect also when there was a well-differentiated tumor. And this is important if we compare with the data that we have from the PDX when we try to engraft the tumor into the mice, because in this nice paper from the Mayo Clinic, you see that the efficiency rate of PDX is about 54% and changes according to the subtype, but above all, it changes according to the differentiation grade. So probably the organoids might potentially fill in that gap of creating a tool for also those well-differentiated tumors. And again, even if the numbers are little, we know that there might be a difference according to the center that is taking the process forward, because really is very highly, it requires a very high level of expertise. So this led us to think that potentially a centralization of the generation of organoid could be useful. And Colin in our team has tried to see if we can derive organoids from cryopreserved tissue. In this case, it would mean that we can potentially test this tool in multicentric clinical trial, because every different institution can just take a biopsy, preserve the tissue, and then ship it to a centralized laboratory. So initially optimized the protocol in a murine model of cholangiocarcinoma. It was developed at the Beatson Cancer Center by the group of Jane Morton. And you can see he has taken, when given the tissue, he divided in 3 different pieces. One was assessed for the normal FPE assessment, and then one piece was freshly processed, and one was cryopreserved with the solution that was developed in collaboration with Vincenzo Cardinale's group La Sapienza. And you can see that even if thawed after many weeks, we can still generate organoids. And interestingly, as you can see in the bottom graphs indicated by the blue and the red lines, you can also understand that the kinetic growth of these organoids are very similar. So this is quite an impactful result. The secretory functions are maintained, so the markers of biliary markers are there. And also, if you look at how the cells grow in live cell imaging, you see that there is quite a good resemblance of how they distribute and grow within the plastics. The genetics, the genomics remain the same. These are data of RNA-seq, where you can see that the majority of the genes are maintained, and even more importantly, the drug sensitivity is the same. On the left, you can appreciate that no matter if you derive the organoids from the fresh tissue or from the frozen thawed tissue, you have very similar response rate in terms of response to the more commonly used drugs. But also, if you repeat this test after you have grown the organoid for a while, you can maintain more or less the same sensitivity profile. So now the big challenge it has is to try and translate these into the human organoids. So unfortunately, COVID had impacted into the possibility to extend the project. So he has already derived a few different organoids from human patients. We have seen a couple of, for example, here I reported one pancreatic cancer and one distal cholangiocarcinoma. We see that these organoids are very similar in terms of genomics and genetics, and above all, in terms of drug sensitivity, you see that they don't really display any difference, so they potentially be used. Obviously, these are just a couple of examples, and then the more data are needed. But I can also anticipate that it doesn't work for all the different subtypes. So I want to just stop here, and I hope that we have raised a few different issues, and I really would like everyone to pose their question and engage in kind of a very informal discussion. Thank you.
Thank you very much, Chiara. Let me just get the ball rolling, and I'm just going to start off from the patient's perspective. if I'm recently diagnosed with, let's say, a cholangiocarcinoma, there are commercially available assays that might tell me which chemotherapy to give and not to give, and they're not always the best assays. So how far are we away from a patient being able to provide some tissue and being able to get an answer on what the best treatment is for them?
Well, in terms of cholangiocarcinoma patients, I think we are still not there yet, for the reasons that I partially mentioned, because we don't know if we start to applying these into clinical practice and we want just to consider and develop organoids for each patient who come into the clinic, in how many of these patients we will be successful. And obviously, as much as we have some evidence from pancreatic cancer field that you can derive organoids from FMB, that becomes more challenging. The less tissue you have, the more challenges you have. And I think that the other big issue we are actually facing in comparison and in contrast to the colorectal cancer field where they are more advanced is that the protocols and the media are not completely 100% spot on. So there is an overgrowth of normal tissues and normal organoids that might impact. And this is why we still need to optimize these protocols and see if we can make them more clinically relevant.
And do you feel that we're going to go to a position where it will be that sort of one-to-one correlation, so an individual patient's organoids to inform about drug? Or do you think patients will fall into strata, so there will be groups of patients who will
Well,
have
the
different
idea
treatments?
is that you can do the one-to-one, with the organoid. At least from Nicola's data, we could see that the negative predictive value, which means, how much you could say that that patient would have never responded to that chemotherapy was very, very high, was greater than 95%. The problem, I think, is understand, if you remember the curves that I shown from that paper are very different from what we usually think when we go for drug discovery projects for active drugs. So the difference in IC50 was about 3-5 times. It wasn't the 100 times that usually you're looking for. So that creates more challenges, I think. If I'm allowed, I want you to ask a couple of questions. One, to look first, as is the expert on the animal models, and with the idea of having immunotherapy now in the management of cholangiocarcinoma, what is your view about humanized animal models? How can we use them, and when do you think they are important?
Yeah, it's working. Sorry. I think actually that's the way we should go. I think one of the concerns that we've always had in animal modeling, both in cholangiocarcinoma and other cancers, is that really the immune repertoire of the mouse does not reflect the human. And so a lot of immunomodulatory compounds, be them kind of like the classical immunotherapies or other things, do work differently in the mouse than they do in the man. There are a number of humanized mouse lines now at the moment, and I do think they're good. I think the problem is that it's that kind of cost benefit thing. They are very expensive. So if you're very interested in tumor immunology, then you should be using a humanized mouse, I think. However, for your day-to-day understanding of kind of cancer biology, then perhaps it's not that necessary.
So probably we are not ready to test the drugs in the animal as well. That would probably
I
not
would
give some predictive.
We also don't know
biomarker.
about the kind of pharmacokinetics of a mouse versus a human often, So if you are required to metabolize a prodrug, the mouse will do that differently to a human. And so actually, whether or not, even in that context, using these mice is appropriate, I'm not sure.
And the thing I wanted to bring in the discussion, Laura, from this morning about your liver tissue slides were quite nice, and I think that it represents a nice potential good model for testing immunotherapy. In one of your slides, you have shown that the cell viability of these models continue to grow. At least at a week, they continue to grow. Do you see that the growth is predominantly from the cancer cells? And do you see that the ratio of different components changes over time that will affect how you can test the drugs?
To be honest, the honest answer at the moment is I don't know. We're still, just exploring that that side of things. We've got some histology sort of done at the moment, and I'll be able to come back to you shortly about that.
Okay, thank you.
Can I ask a question about that as well? Sorry.
So Laura, if you remain at the microphone.
Sorry. It's like musical chairs. So within those slices, not necessarily the work you've done, but perhaps other people from other cancers, are the immune and stromal components maintained in their kind of normal state? Or, I suppose immune cells are quite plastic in their phenotype. So do they repolarize? Do they change? Or do they stay fixed?
To my knowledge, and I'm not an immunologist by any stretch of the imagination, there are some changes initially because it is obviously a very stressful situation that we put the slices under, but they do recover over time. I am aware of that. Yeah, that's the limit of my knowledge at the moment, to be honest.
Easy.
Hi. So actually, I had a comment about that because I found Laura's tumor slices very interesting. So I did my PhD in melanoma immunology, and we worked on tumor slices. And what you do find is when you slice through the melanoma and just put it in plastic on certain media, you do get crawl out of immune cells. So T cells, B cells, fibroblasts, dendritic cells, they crawl out from the skin. And we would keep these slices for about a week. And when you phenotype fresh slices versus slices that have been in culture for a week, you do see quite a change in the immune component. Granted, a lot of the stuff we looked at was sort of phenotypic. You can look at functional stuff to a certain state, but lots of immune cells downregulate their receptors when they're in plastic because as a way of conserving energy. So I think in terms of testing immunotherapies, I think that's something we have to be a bit cognizant of. You're going to have differences in the immune cell milieu from the fresh versus the one
And in
that's been
that
there for a while.
situation, what about the cancer cells? So one of the big, well, perhaps one of the early concerns with organoids was that you were going to get essentially like huge amounts of genetic drift and kind of like really strong selection pressure for mutations that enable the organoid to kind of grow in a dish. So did you see that in the kind of melanomas as well? Do you get selection of like BRAF activation and things?
So we didn't look at the tumor cells in terms of the mutational drift, but there was what's her name? Professor Sophia Karagiannis was quite interested in B-cell antibody responses to different types of melanomas, and she did see very differential responses in what classes of antibodies you had being produced and responding to the tumor. And I think she did some work linking that to changes in antigen expression. So again, whether that sort of links to mutational drift, I can't say.
Yeah.
But definitely, that was the data she showed.
And Izzy, can I take advantage of you while you are at the microphone for your pathological view? And probably, I don't know, Tim maybe can comment as well. I mean, for developing organoids, if we consider about a biopsy, then it's going to be an extra core and, we are going completely blind. But if we want to apply that to an early stage, so we have a resected specimen and maybe we have a little extra time to develop the organoids and then, inform the adjuvant treatment. So do you think that, I mean, we have shown it's feasible, but do you think that is applicable to all tumors, or you think that as a pathologist you have a difficulty to really give a piece of tissue for each patient without, limiting the possibility of having a proper, fully diagnosis with assessment of the margins?
I mean, definitely, it depends on the size of the biopsy, because if you get really tiny pieces of tissue, then unfortunately we're just going to want it all for diagnostics.
No, I'm talking about the resected specimens.
In the resected specimens, definitely, because they're quite...
Even if you're talking about perihilar cholangiocarcinoma or distal, where the sites might be smaller and the margin might be more difficult to be assessed. Do you see any challenges there?
I mean, so from my experience, the resection specimens are either you get a hepatectomy or you get a Whipple's resection. And even if you have a hilar cholangio, you will still get good amounts of tissue within that. I think the key thing, and again, I was really impressed when that the surgeon was being allowed to take the samples. It means they trust you. Because we're really finicky about who takes the specimen, because we have to prioritize
Yeah.
diagnostics. But if you're careful, you can get tissue for research. And I'm really thrilled to see the fact that you can freeze the sample and still get it out later to develop organoids. I think that's amazing.
Yeah, that is remarkable.
yeah.
I think so if you've got a tissue sample that you freeze, so your fresh one, you get organoids from it, and it works great. Does that mean that you will absolutely be able to get one from the frozen? What's your recovery rate from the frozen?
Well, we don't have a lot of numbers to really in human to really draw the conclusions. What we have seen so far that if you don't grow from the fresh, it's very unlikely that we also grow from the frozen.
Right, okay.
I mean, it might be kind of a sampling process, but the couple that we have done didn't grow from the fresh and not even from the frozen tissue. We have had a case where we were able to grow the organoids from the fresh, but not from the frozen. But what we have actually seen, and I think this is becoming a more solid trend, is that, for example, ampullary intestinal, they don't work at all. And it's easier to develop from the fresh, but from the frozen, no. And the protocol, the cryopreservation solution, has been actually targeted towards large bile duct cells. So maybe this might explain why it's more successful in pancreatic distal bile duct, and we've had one success with an intrahepatic cholangioma, as well as some HCC murine models, for example, seems to be good, but it doesn't work, in the intestinal phenotype, for example.
Interesting.
Sorry, it was just is that working? It's just a point really to follow on from Lauren, the question you asked. So, slices as a platform really to modelling immunotherapies really be dependent on what the resident immune cell populations within that slice that we've taken are, and that's sort of where we've recently received some funding to take that work forward. So what we want to do is look at what the immune phenotype is within our resected specimen, and then obviously then within the tissue slice and what happens to that over its time in culture, and then obviously whether or not that could be modulated, and again what the response would be to immunotherapy drugs, so like sort of PD-L1 blockade or something like that. So so we're aware that sort of that's where we want to take things, and those are some of the answers to that question. I actually had a question for yourself, if that was all right. I mean, I don't sort of really know a huge amount about sort of animal models, certainly from that, but obviously when you're looking at modelling sort of response to immunotherapy, you want an immunocompetent host. So is there a way to get sort of, we talk about PDX models, to get human tissue from, so say from theatre, get that into an immunocompetent mouse? Is there a technology to do that? Is that something which in the horizon is going to be possible? How is that feasible?
I mean, at the moment, I don't think it would be broadly feasible, because obviously you're going to have a T-cell response to your your grafted tumour. Yeah. With things like the humanized mouse, so there are various ways to humanize a mouse, and so one of the easiest ways is to irradiate the recipient mouse, transplant human bone marrow into it, and essentially allow its kind of immune repertoire to be humanized. And in that context, you can grow grafts, but they don't grow well. I mean, there's still something there that is kind of rate limiting, I guess. It's still very much you put things, these kind of, I mean, if anyone else knows different, please say. But as far as I know, you kind of still put these kind of xenografts into nude mice, or athymic mice, typically. Izzy.
So again, Professor Karagiannis, who I worked with at King's, she did some really nice work where she was essentially able to create humanized melanoma models. So they would, like you say, irradiate the mouse, reconstitute it with a patient's PBMC, so blood cells, and then implant that patient's human tumor into the mouse. So you essentially had the patient's immune system and the patient's tumor in the mouse, and she used that to show quite some astonishing work on the antibody responses, the B-cell antibody responses to melanoma. Like you say, it's prohibitively expensive. I mean, we're talking, this was like 2013. Hopefully the costs have gone down now, but again, it's a really finicky process to get set up as well, but it is possible.
Yeah, absolutely. And I think one of the rate limiting steps is that you do need then, enough material to reconstitute the mouse.
Yeah.
And timing-wise, you've got to be really spot on, and it requires you to have excellent surgical collaboration, I guess, right? So, So you can all coordinate it.
And again, in melanoma, you get a lot more tissue than you do in cholangio, even with biopsies. So.
Sorry, I was thinking, I don't know, I know it's probably not the best solution, but using, for example, syngenic models would be also an approach to test immunotherapies, no? I mean, there are already several mouse cholangiocarcinoma cell lines that can be implanted in immunocompetent hosts. I know that the immune response is not the same as in humans, but I guess it's the easiest way and cheapest now to do it. I don't know if it would be possible comparing with the humanized mouse models.
Yeah, I mean, I agree. I think there's so the nice thing about a syngeneic mouse model, so syngeneic model is where you take essentially the same background mouse cells and put it back into the kind of same recipient, so you don't get an immune rejection, is that you can take those cells out and edit them or do things to them in vitro, right? So, and then put them back into the liver. And I think that has a massive benefit, even beyond kind of, what we do with the transgenic mice, which are still very, very expensive. If you can get to a point where you have a Black 6 mouse line that you can edit and then put straight back into the bile ducts directly, I think that'll be very cool.
And what do you think about, I don't know, it's totally hypothetically, implanting in immune deficient mice CCA cells, human CCA cells with CAFs or TAMs, for example? So do a pool of cells and implant them in the liver of immunodeficient mice.
Yeah, I think that'd be awesome. I think this is again kind of one of the big limitations of when we do transplantation models or kind of even kind of classical xenografts, is that we only ever put in epithelium. We don't really consider combining the stroma and the immune repertoire. I mean, to that point, what are the developments of modeling the microenvironment in the dish?
Well, there are some successful data about co-culturing the organoid with CAF endothelial cells, immune cells, to a level, for example, in colorectal cancer, they've been able to see that mismatch repair tumor versus mismatch repair deficient and proficient tumors. Organoids actually, when they are put in co-culture with these cells, for example, they are really behaving differently. The T cells can kill the deficient ones. And I think that now with the 3D printing, there is the possibility to recreate the bile duct in some way, the liver, and then you can put the organoid inside. I think that probably still a lot to be done. And I think that, we just need to move forward with the development of organoids to really see how that is impactful. Yeah. I mean, with the syngeneic mice, in answer to you, Pedro, I think that absolutely is a valuable model for biology. If we want to try and see kind of a, how can I say, maybe dreaming future of individualizing treatment, then probably, it would then be possible to individualize the treatment on that patient, use the patient's tissue.
Yeah. Sure.
Hi, I just wanted to make a comment about making PDXs with tumor epithelium and fibroblasts and other tissues. I think that's what Anna Grabowska is doing at University of Nottingham, and she's got very good results with that. And they grow well and they resemble the primary tumor really well. So I think that technology is up and running at Nottingham at present.
And I think in that context, you're kind of overcoming perhaps some of the issues with having to humanize a mouse, right? Because you're putting, like you say, the microenvironment together with it. So you may, not have that problem.
Can you just come to the mic? I'm sorry, just for the people online. Thanks, Izzy.
Sorry. I think the only drawback with Anna's model is that you don't get the immune cell representation within the PDX
the immune
spice.
cell,
Yeah.
So you don't get the immune cell representation.
Yeah.
But you get the
Well,
CAF.
you get the CAFs
You do.
and the stromal supports.
Right.
And her PhD student has shown quite nicely that in terms of the histology, the tumors look the same as what you get on the H&E when we do the fresh tumors.
Could I just ask a sort of probably a bit of a stupid question, but other than mice, you mentioned, Luke, that the immunological component in mice is quite different from humans. So even if you can get a syngeneic model working really well, you might still not get a very good answer for how the immune system is working with the tumor that's growing. So I just wondered, does anybody know of another animal model that's more representative or more closely similar in terms of their immune system than mice to humans, if you see what I mean?
Well, I
Like
think
syngeneic, anything else?
I mean, if we think about the virology, they have used the non-human primates for HCV modeling, which I think in some way could better represent more similarities. But it becomes kind of a very difficult
primates,
to manage.
Are pigs at all similar, or?
So
I just wondered
I
because
don't
I
actually
noticed
know.
you had pictures of pigs
I'm
up there.
I'm not too familiar with the oncopigs, and I don't think that there is the whole immune system. But I mean, there is the immune system, and definitely is more similar to the human. I think that what a recent paper has shown is that they've been able to reproduce the NASH background in a better way compared to some other rodent models. And but so far I don't think that there is a lot of knowledge about that. There's been some very recent data about it.
Okay, thank you.
Thank you very much. It's been fantastic sitting in a lab meeting, just hearing how these guys collaborate and develop ideas and collaborations. I think it's been a fantastic session. Thank you very much to Chiara and Luke.
Thank you.